Good evening, everyone. Okay. So yesterday, we used plot function for visualization, data visualization. We generated different types of plots. Okay. So let me open. Yeah, yesterday's part. Yeah, here. So these are basic plot function we used with single data set, multiple data set, and pie chart, 3D pie chart. Then many more plots, like here you can see line plot with line function, box plot, histogram, bar, pie chart, even area plot we can plot here, dot plot, density, and correlation. Okay, and we just checked how to use ggplot package and how to uh, apply different functions from that. Here we fetched data set, that is cars inbuilt data set in R environment, and iris. So let's explore more about ggplot with different types of plot we can generate here. Let me first call this data, and we need library as well. Okay, so now we are ready with iris data and ggplot package. So as we have yesterday, we plotted here point plot, that is the first ever plot, very simple one, and that is called as a scatter plot as well. Scattered data points are there. Okay, have you tried this? This is scatter plot. Now let's use some different types of visualization with the same plot. We can just edit here. So rather than scatter plot, with the same data we can generate a bar plot. So bar plot for data, that is iris itself, either you can provide like this, data is equal to, means data parameter, and then assign iris as a database. This is also okay, otherwise you can just write iris because this is already downloaded in the environment. Then for bar plot, what you want to plot, like for which bar plot, uh, for bar what you want to set, that you have to specify here. Like first thing, aesthetics we have to provide. Aesthetics means for x-axis what you want to feel, what you want to provide, like species is there, then sepal length, petal length, many parameters, many columns are there. So let's take any one of this. Or if you want to generate a bar, better we can generate for species. So here I am fetching species column, and also color. Now color is what? So color is basically that bar for on the basis of species that will fill. And then here we need to change function. Rather than geom point, we can set this as a bar. That's it. Here also title you can change. Title, x-axis label, y-axis label, these all things you can change. So oh, I'm changing in previous. Okay, fine. Or let's change the name here. Okay, so this is a bar plot, and you can specify like on what basis we are generating that plot. So this is nothing but species count. As we know, there are total 150 rows, means three species are divided. These are three species. So let's see how they are divided. For x-axis we can specify here, this is nothing but species, and for y-axis is your count for that species. Okay, that's it. Theme minimal is fine. This is standard theme, and execute. Okay, here we have not colors on colors. Okay, sit, but they are not appearing properly. So let's check somewhere we made mistake. X is species, but we are going to fill on the basis of species. So here I'm writing fill species. Now it will work. Can you see here? For three species, I think for each species they are given 50 count only. That's why it is showing the same count for each species. So this way you can generate bar plot. This is the original one, scatter plot. Now we are going to create this one more plot with help of geom function. So here let's generate box plot. So box plot is, as you know, this is for statistical calculation. Okay, means you'll get a median line, you'll get a first, second quartile, then whiskers. These all things are there. So for that, let's provide the details. Iris dataset we are using. Aesthetic sepal length is fine, or if you want to change on what basis you want to generate that box, that you can specify here. Okay, let's see. Three box we can generate. So I am taking your species. Okay. Then for y-axis, let's take sepal length or width, anything is fine. And you want to create box, so box of you can use here fill. Fill means what? Fill color on that particular column. So here we can set fill for species column, means that color appears for that boxes according to the species name. Okay, so here just need to change in box plot fill on basis of species. Remaining thing as it is, change the function rather than point. Now we are generating box. So here the function is box plot, not only box, but that is box plot. Then title, this is a box plot. If you want to specify the box plot of sepal length by species, then you can specify. Okay, then let me take this on next line. X-axis is here, we use sepal length. So take this, and y-axis. But here we need to change this. Box is like x-axis shows the categories, so we have placed here species information, right? Let's check the example of this. Minimal is fine. Staple length not found. Okay, there should be L capital. Now you can see the average length you'll get here for sepal. This is, I think, minimum length for Setosa variety. Versicolor is some higher than Setosa, and Virginica having very high length for the sepal. So accordingly, you can see the median line for three boxes. Clear? So here species is basically, it treats species as a categorical variable. So here there are three categories. So that's why we have created three boxes. Okay. Then y means how we are going to map sepal length to the y-axis. That's why we have plotted that sepal length. You can try this with petal length. So you'll observe these all things, and also you can save these all plots if you want to study on that particular topic, like varieties and their sepal and petal length difference. So statistical summary you'll get for each and every column, and you can export this and then make a combine chart. This is good for visualizer publication. Okay, so now one more plot we can create with existing data set, that is frequency. We already created bar chart, but now we are going to generate a frequency with help of histogram. So for that, what we can use geom histogram, and also in histogram we can use different parameters, like either we can set how many beans you want. Then say, you can set width of each bean, even position of that bean. Position means like side by side you want, or how, what exactly you want. So these all things are there. So let's create one histogram with help of geom for the same data set. Why I'm using same data set? So it is easy to understand for the same column how different graph we can generate, and how to check result, analyze the result. Okay, so this is for hist or histogram. Data is iris itself. Then what you can use, your sepal length you can keep. And for histogram, we'll fill with the species itself, as there are three species, so it looks good. Change the function. Now inside this function, we can add different parameters. Like as I told, we can add bin width. There are bin. You can see here different functions are available. Okay, let's decide some width. I'm taking minimum, and also there's a position we can set. So there is one option. Okay, now title you can change. For sepal length, by species, fine, and then length. Now here for x-axis, this is length. So I'm just changing this. And y-axis, what will be for y-axis, as this is histogram? So just tell me, because we already created histogram with the simple plot function. So whenever we are plotting histogram, what what will be on y-axis? If sepal length we are using,
It is the frequency generally, so maybe
yes, correct. It is frequency. Okay, so here we use bin width and position as a parameter in histogram function, and we have generated this. Just let me execute and check. Okay, so for three different species we are getting this histogram. So by observing this peak, you can see the top, the peak is 5, color is red, setosa. Okay, so highest peak is for setosa. So same type of graph we can generate, like rather than these bars, we can create a curve, like this curve. Okay, we'll see that as well. That's called as a density plot. So for density, let's create here. So we can set the opacity, like by using alpha parameter. Transparency level we can adjust. So call ggplot function. Inside that, first we need to provide iris dataset, then aesthetic. Inside aesthetic, for here we can use, let's use length itself. We are focusing on length, so let's check length. So this is length for x-axis. Even we can decide color for that curve. So as there are three different species, so we can differentiate these on basis of species. So color I'm using here for species, and feel that color by using species information. Okay, that's it. Then here you need to provide plus sign, then add one more function, that is geom density. Let's set transparency by using alpha. I'm just copying this label and everything. Okay, rather than frequency we can change your density. Okay, remaining I'll keep as it is. So here the same picture we can see in different format. So the three color and this opacity, you can increase the opacity, like suppose here I'll keep 0.8, like this. Transparency level, depending on your choice, you can keep. Now the one more graph we can generate, that is violin graph. So in violin, suppose we have created the, or let me copy this as it is. Okay, need to check because this is iris dataset. Aesthetics we can use for violin, because we violin is just a form of bar plot. This is showing bar like structure or categorical value you can use here. So this is species. Then why we can keep sepal length and field species fine. Just need to change this. Okay. If you want to trim like true or false, these two options are there. It depends. Let me check this. Okay. But for trim, yeah, we can set trim as false or true. So that basically ensures violins are means that that shape or that uh, bar type structure is not trimmed at the edge or data range. So for that we can set here false. Okay, and we fill with the species. So it will, the plot will generate by the species, three species. So three violin like structure you'll get. Not exactly violin, but yeah, you can consider. Okay, so you can also replace whatever default color with manual. Like suppose you want to scale fill manual, that parameter you can use, and you can create your own palette. Like suppose three different colors you want for these three species. So you can create, let me show how to create. You can create your palette. Like suppose you want to create one palette for these three. So you can set one variable like this, any variable name you can give, and then here you can provide, no need to use plus, values, and create a set or vector of three different colors. Like here you can mention red. If you want whatever color you want, that you can mention. Like this you can set, create your own palette, and then you can use this variable. Even there is one more option, that is faceting. Faceting means suppose you want to create a plot by using another variable, means separate that particular variable. So there is one more option you can use, that is called as a facet wrap. Facet underscore wrap. You can read more about these all things here. Suppose on the basis of species you want. So for that, like species wise, you want three different plots. Like here somewhere in density, no, in here we generated histogram, right? In one plot only there are three merged species and their bars. So if you want to generate three different species and three different subgraph, then you can use on what basis you want. Suppose I want on basis of species column. So tilde sign you can use, and you can write it like column name. Also, here we use theme minimal, but yeah, you can try different themes. Like there are many themes. You can see here theme. You'll get gray theme, minimal theme, classic theme, dark theme. So anyone you can try. Okay, so this is all about ggplot. Okay, you can try with gene expression data. You can generate uh, different kinds of plots. And there is one more topic we need to cover, that is statistical test. So are you aware about any of the test? Statistical test like t test, z test, or chi-square test, ANOVA test? Any idea about this?
No, ma'am.
Okay, fine. So let me tell that part. First, let's understand what exactly is statistical test and why we are using this. Are you aware about term p-value? P-value?
Yes, ma'am.
Okay. Because p-value is a major parameter to read and to understand the result of the test. Okay. So first, let's discuss about statistical test first concept. So t-value, there are four type of tests we are mostly used. First is t-test, then z-test, there is ANOVA, and chi-square test. Okay, so t-test basically used to compare mean of two groups. So that two groups might be two samples, like normal sample, control sample, disease sample, like this, or where two groups are there and minimum quantity is there, like less than 30 elements are there. So we can prefer t-test. Means if they are statistically different from each other, then by observing mean of these two group we can understand or interpret the result. So this basically used for gene expression studies, where we need to check expression level of the gene in two different conditions, or we just want to compare the conditions. Okay, so small size, we need to prefer this t test. Then there is a z test. So z test is for bigger size data, or we can compare this different samples and population mean to determine if there is any significant difference or not. So when we use this in bioinformatic application, like sample size is very big and we have population parameter, that is st, standard deviation, which is already known. So that time we can prefer z test. Fine. Like suppose for example there is a two countries we are comparing, means there is a huge data and we are comparing average height of let's take men or women in these two countries. So what exactly the significant difference, to check that we can prefer z test. Okay, then there is a ANOVA test. ANOVA stands for analysis of variance, means this is basically used to compare means of uh, three or more groups, means it is greater than two groups we can say. So this is very useful when we want to compare or when we want to check experiments with multiple conditions or multiple treatments. Like suppose there are multiple groups and in that group we just want to study gene expression, like there are 3 to 4 samples and we want to check what exactly gene, which gene is expressed, like that. Okay, so multiple treatments, multiple conditions, when they are prefer ANOVA test. For example, like a simple example which can relate with this gene data, that is I can give. Suppose you want to compare diet plan, diet on weight loss with several groups. Okay, different groups like age group you can consider, or male female, like this you can consider. So multiple conditions with multiple set, like this. Okay, and there is a chi-square test. So chi-square test is basically when there is a significant association between two categorical variable. That time this test is mostly used, like in genetic association study, where you need to check frequency of alleles or just want to check genotype which is compared between two different populations. So this time chi-square test is preferable. Means if you want to examine a relationship between variables, um, just want to study like uh, what exactly the link between smoking status for lung cancer, like this. In that situation, you can prefer chi-square test. Okay, and significance, null hypothesis is term also there. Null hypothesis, what exactly null hypothesis? Any idea about this term? Null hypothesis basically when means what you can say that nothing special is happening. Okay, means if you are testing any new drug, and null hypothesis would be like that drug has no effect on patient compared to like condition. Okay, and alternate hypothesis is what? There is again a test which is for alternate hypothesis. So that resist, or that can be, you can say, effect of other means difference. So these two conditions you need to check. Also p value. P value, if it is low, low means you need to compare with the standard, and standard is what? Like mostly compare, it should be less than 0.05. Okay, and if it is low means typically less than 0.05, then it suggests that results are unlikely to have occurred by chance. So you may reject the null hypothesis. And conversely, if there is a high p value, so that result what suggests? They are more likely due to random chance. So you may not reject that null hypothesis. So these two important terms we need whenever we are going to check this result. Okay, so let me open. Or as we don't have data, so what we can do, we can simulate here itself data, as we know how to simulate data. So let's consider first t-test, and for that we can generate here itself two data set. One is control and one is treatment. So for control, let's use function or norms, and here it is for lesser values. So 30, minimum 30 we need. So we need to provide your mean, standard deviation. So I'm providing mean, and SD we can keep 2. Okay, so this is the first sample. Could not find function or, yeah. So you can check control, 30 data is there. Similarly, similar way you can generate one more data set for treatment. Okay, so this way we have two data set. If you want to change something like mean, we can change. This is one data. So we have now two data set. So what we need to do, we just need to call t-test function and provide this two data set. Or we can create one variable to save this result by calling t-test result, and then call t-test function t dot. Here you'll get tests from stat, then provide these two set, control, comma, treatment. Okay, execute, and then call just result to check all parameters. So here this is a list form. Okay, execute this result. Okay, so what all things are there? You can see Welch to sample t-test. Okay, so here you can see different parameters. Data we use, that is control, treatment. T, that is t statistic, means what? T value, that is nothing but a measure of how far the sample mean are from each other, and that is scaled by variability in the data. So here is the variability, and how to evaluate actually this value. So larger absolute value if you are getting, so that indicate it is stronger evidence against the null hypothesis. Then there is one more parameter you can observe, that is df. This is called as a degree of freedom. So degree of freedom basically reflect the amount of information which is available to estimate the population parameter. Fine. So here this is from Welch t-test, which adjusts this df for unequal variance and also for sample size. So when there is a higher degree of freedom, so typically you can say mean are more reliable result. Then there is one more parameter we can focus, that is this confidence interval, means ci. Okay, so range of values within which that, if it is true difference in mean, so that likely to be a lie. And if the ci does not include 0, then it supports a significant difference between the mean. That is the thing. There is also a alternate hypothesis, you can see the true difference is mean is not equal to 0. Okay. Then sample estimate, that is mean of first data set, this data set, what values of this control, and second data set. Okay, so here we are getting mean which is higher in second data set. Okay, so when we compare the mean to understand what exactly the direction, what exactly the magnitude of the difference, so first we need to focus on p-value, confidence interval to determine statistical significance, and then you can use sample mean and confidence interval to basically assess the biological relevance in the result. So simply the same thing we are going to use in z test, but we need a large data set. So what we can do, we can increase the size. Let's take 100. So z test is similar to t test, only difference is what sample size is large, greater than 30. Okay, means, and also population variation should be known. So we need to write that as well here. Not changing any other thing, just change the size, or we can change the name. Okay. So now here we need to call z test function and then provide these two variable which we have created. Along with this, you can also add two parameters, that is sigma x and sigma y. So sigma x is basically for first set, and y for that is for standard deviation. That, okay, just let first call this z test, or in here itself I'm adding. First we are going to add sigma x, sigma from stat for first data. Let's keep this 2, and similarly sigma for second data set is 2. Okay, so this basically conduct comparing between two groups, as we have these two groups. Similarly, control, treated, any two group which we are using. This is not showing. Okay, for z test we need library. I think I have not initiated that, just let me check. We need library called as bsda. First you need to install that library, as I already installed in my environment, so I'm just activating that by calling library function. Yeah, now you can see this belongs to bsda z test function. Now it is working. So this group 1 and group 2 means our control and treated. Mostly when we are working with GWAS data, GWAS is what genome wide association studies. So that time we can prefer this Z test. Means we can observe allele frequencies between different cases or control. differ significantly. Okay, so let me execute result. So same parameters are there. Here two sample z test, p value which represent your probability for difference, which is, if you can check here, it's z value is also there, alternate hypothesis, and the same parameters, but need to check the mean. Here we keep 10 and we are getting here 10 and 11. Okay, so average value for each group is nothing but that mean. And when we are using a population mean, just want to calculate or estimate the population mean, then we can check this. And accordingly, here if we compare the mean to understand the what exactly the direction or size of the difference, then you'll get here which group is higher. Treatment group has higher mean than control. Okay, so first thing, p value, which determines if the difference is statistically significant. Then focus on confidence interval, means basically it confirms the range of mean difference and also exclude zero for significance level. And finally, sample mean and z value, that helps for strength and direction of the difference. So this is the thing. For ANOVA, basically ANOVA used to compare mean of three or more groups. Just want to check or just want to see if at least one group mean is different from other groups or not. So for that we are using ANOVA test. So ANOVA, for that also we need data set. So what I'm doing, let's work with small data, but we need more than two data. So here you can add one more data set, like this is a control, this you can consider treatment one, and similarly create one more treatment. Suppose we are giving two treatment to any sample. So like this. Okay. And if you want to change mean, then you can change like 10, 12, and here we can keep 14 by difference of 2. Let's execute one by one. So these three data set we are ready. Now time to perform ANOVA. So for that, call function AOV. Here you'll get a how to write this. So first thing, here you can provide a means compare mean of two groups or more than two groups basically. So let's first mention values. It is given here. No, the syntax, let me check first. But for that, this is a syntax, basically values versus group. But for that we need to create these all things, means in table form we need to create. So tilt sign is there, and which data we want to use for that, that also we need to provide. So data is nothing but, let's create first data. So to create data, I'm writing name data itself, so no confusion. And we need to convert these three samples as a data frame. So for that we need function data frame. So data frame is nothing but collection of vectors. So here you can add vectors, and first vector you can keep this value. Okay, so value is nothing but what, how many groups are you have. So as there are more than two groups, more than one group, right? So you can create a vector to save all this group name. So suppose first is control, second is treatment 1, and treatment 2. So these three groups we have. This is first vector, means first column of this data frame. And then you can create a group. Group is also for, a group is basically for categorical data, and category we can decide by using, there is one more function, if you remember, we discussed in data type, that is factor. Okay, factor is for categorical data. So here you can call factor, and as this data, like there are total 30, 30, 30 means total 90 elements are there. So if you want to provide the name for this data frame, like for these three um, elements, okay, so you can use repeat functions. Like for control, you can provide control name, treated, and treatment 1, 2, like this. So here I'm using repeat function, and repeat we can use for both data set. This is name, so you can keep right as you want, control sample or control. So this is first treatment and second treatment sample. Now how many times you want to repeat that name? So that also, because in repeat there is one parameter each, if you remember. So each there are 30 elements for each sample, so we can keep this 30. Okay, so that's the way how we can generate a data frame. Okay, I've missed that bracket. Okay, so it is working. Some, I think I missed that bracket. That's why it is the showing unexpected symbol. Fine. So here, let's check what exactly data frame we created. So for that, it's 90 rows. It's difficult to observe. So better we can call head function to check data. Here you'll get, so value and group, these two vectors or these two columns we have, and these are the values. So by using head, we can see only first six values. If you check bottom to check total count, so for bottom we can call tail function and provide same data. Okay, so here last row is 90th. Okay, so and the first, initially it is start with control. From bottom, if you can see, there's a treatment too. So now this way we have this data frame having values and group. So we can use this here to calculate ANOVA, or let's create one variable to save that result better. That is ANOVA result. So as we have given name data itself, so let's provide data. That's it. Values. What is this value? Now either you can directly check ANOVA result, or you can call summary function to check ANOVA result. Okay, let's execute the result. Can you observe here something? Star, star, and all. So, what exactly this star and how to read on this actually? So, ANOVA is basically, suppose, let's interpret this. Group means what? Group basically reflect number of group minus one. How many group we use here? Control, treatment one, treatment two means three groups. So, three minus one, how many group we have? Two groups. Okay, similarly residues as well, we are getting here 87, means total there are 90 rows, 90 minus 3 samples, so we are getting this 87 answer. Okay, so group, that is three groups minus 1, and also for residues whatever data points we have generated minus 1. Now between group means number of group minus, like if you consider any number, suppose here we use 3, but if you consider any number like 5 groups are there, 10 groups are there, so groups is always number of groups minus 1 is the value. Okay. Mhm. Now when you check degree of freedom, this DF stands for degree of freedom. This is nothing but sample size and grouping structure, like how much information is available for analysis and all these things. So this is the same thing. Now there is one more thing you can observe here, sum sq. That is nothing but sum of squares. Means between group and within group, both. Okay, between group means treatment, like variability due to difference in group mean, and within group, that is nothing but residuals. That is a variability within each group. So either that is error or that is noise, anything. So if larger between group sum of square, you can compare this with within group. So that is such as that group differs significantly. Then there is a mean square. Mean square is also, if you want to compute that mean square, you can use sum of square divided by different. So this calculation will give you this count. So this is for comparing two mean squares. Then f value. F value is nothing but ratio between group mean square to within group mean square. So these are all statistical things. Maybe it is difficult for you to understand, but yeah, just one thing important, that is PR. PR is p value or probability of observing an f value. Okay, so if your p-value is less than 0.05, you can reject the null hypothesis. Just remember this thing. And if your p-value is greater than equal to 0.05, so you fail to reject the null hypothesis. Means there is no significant difference between groups. And these score basically 3 star, 2 star, 1 star is what? So these are nothing but corresponding p-value threshold. Like for first I'll tell this 3 stars. 3 stars for p-value means it is less than 0.001. The value is given. So significant. Like significant level you can understand by observing this star. Okay. Clear? There is a chi-square test as well. So first understand what is the chi-square test. So when you want to check significant association between two categorical variables, that time you can prefer chi-square test. Like suppose you want to analyze categorical data, like for example snp snp association study, and where you want to check, or if you want to just observe genetic variance is associated with that particular trait or that particular disease or not. So it basically help to determine if that distribution of allele difference between cases or difference between controls, like that. So for that, let's create one example for that. Suppose we have one, we need, I think, matrix. So let's create matrix first. For first observed data, matrix function we can apply how many elements you want to create. If you want to create a random elements, then you can use random function. Otherwise, just provide vector of few elements. So let's provide few elements here. Suppose I'm writing here 4 elements. So four element matrix means you can provide your number of rows, and if you want to read that data by rows or not. So I'm writing here, yes, number of rows we can write 2 by 2. So let's provide 2. And by row, if you want to read your data by row name, then you can provide true, otherwise false. So this is the first thing. You can call this to check. Okay, so these are the values. Now let's provide column name because it is a matrix, so there is no column name, row name. So call names function we can call, and for this data set we can add column name. As there are 2 columns and 2 rows, so only this much is required. So provide 2 names. We can provide, suppose we are looking for 2 population. This is one name. Okay, this way. Then check this observe. So yeah, column name is added. Similarly, we can add row names, as we have keep here row name by row true. Okay, so rather than call names, we can call here function row names and change the name. For population, we can keep here, suppose we are looking for mutant versus wild type population, so that we can set the first is for mutant and second wild type. Now, how to interpret the data or how to call first function. So for that, there is a test name, chi-square test. Here it is, last option, and provide the data set. This is our data. Or let's create a variable. Now here you can see Pearson chi-square test with the correlation they have used. Internal algorithms work for this. Okay, so if your p-value is again significant, significant means less than 0.05, so there is significant association between SNP type and population. So you can check residues for specific cell which contributes for this association. Okay, so here what we are getting, x squared, df is again degree of freedom, p-value. So x squared is what? X square is basically chi-square statistic, which measures how far the observed data uh, from expected data under that null association. Basically, means there is no association. Okay. Remaining thing we already discussed, like what is degree of freedom, p-value. There is one more thing, this correlation, the title which is given in your correlation. So means correlation basically when we are analyzing here our data is what, 2 by 2. 2 by 2 is a table like matrix, and which we want to reduce bias due to small sample size. So when you want to ensure more conservative test results, like small sample size to, if you enlarge the data set, then there is a correlation, you will get this. Okay, now basically these all tests are you can use in biological application. Suppose you want to check, or you just want to do hypothesis testing, or want to do data interpretation, any statistical significance, so that time you can prefer this all kind of test. Okay, so yeah, this is our last day of R module. Okay, we have completed R module, also Python module. So in R, we covered this data manipulation, data visualization, some statistical techniques we covered. Means from simple plotting to advanced analysis, advanced visualization. So I hope you have developed a skill that is essential for your field, bioinformatics field, or if you are more interested in these all technical things, then definitely there is a broad spectrum for you. Like in data science, you can explore. Okay, so keep experimenting and let R help you to turn this data into impactful insight. So best wishes for your future and keep coding.
Thank you.
Yeah, all the
Thank
best.
you, ma'am.